All right, this is going to be the final section for this exam. I am sorry that I have been posting this extremely late. I have been dropping the ball, so let's get into this. I know you guys are waiting to go into the material, and it's very confusing, but I'm hopefully going to be able to break it down to make it a lot easier. And this is going to be all in one run through. So going into lecture 10, ETC, oxidative phosphorylation. This slide is just generic stuff about the mitochondria, how it's introduced into the cells. Realistically, you may not even use this on the exam, but it's just basically the mitochondria is uptaken by the cell, the prokaryote, and it forms the eukaryote. That's it. But on the MCAT, this is good to know. So I just put this for your reference. So just some general stuff about the mitochondria. The mitochondria is a double membrane organelle, which just means that there's the outer layer and then there's the inner membrane, outer membrane, inner membrane. And these are going to separate the mitochondria and what we call in three compartments. There's going to be the cytosol, which is just the outside. It's going to be the intermembrane space in the middle, and then the mitochondrial matrix. So it's good to keep these terms very clear. And if I ever misspeak, just know that I misspoke ever in the future. So we know that glycolysis is going to occur in the cytoplasm, and we know that the end product of glycolysis is pyruvate. What's going to happen with pyruvate is that we know it's going to turn into acetyl-CoA, which is going to get introduced into the Krebs cycle. But where does that occur? That transformation from pyruvate to acetyl-CoA, it occurs inside the mitochondria. So the pyruvate doesn't just freely diffuse through because the inner membrane is not permeable to any solutes. It's impermeable. So the pyruvate has to go through a protein channel, a translocator, and the way it does so is through this symport with protons. You can just call it like a pyruvate symport. And essentially a pyruvate will go into the matrix along with a proton as well. And just like a little sneak peek or teaser is that with the proton motive force, we want to build up a gradient inside the intermembrane space, so we don't want the protons to go into the matrix. So that's something to keep and distinguish is like the symport, it basically decreases the proton motive force because the protons are able to freely go through. And then ETC, that's all I need to say. So talking about building the proton gradient in the ETC, we're going to basically give just a rough overview before we go into all the complexes. So what I wanted to mention is that as the electrons go through each complex right here, we're going to see the protons go into the intermembrane space and pass through the inner mitochondrial membrane. So it's going to go through and basically form positive charges just around the mitochondrial matrix, around this inner mitochondrial membrane. So you see the pluses on the outside. What that means is because the positive is leaving the cell, we're going to form a negative charge just because of the absence of the positive, if that makes sense. Because there's no because of H protons are leaving the mitochondria, we're going to form this dipole of it's more positive on the outside than the inside. Therefore, the inside is more negatively charged. We're not adding more electrons. It's just the fact that the protons are leaving that's causing this negative charge to form inside the mitochondrial matrix. And so you can see this distribution or dipole, you could say even, of the pluses on the outside and negatives on the inside. And then eventually that's going to drive this proton motive force because the protons want to go towards that negative charge, right? But it can't pass through freely through the inner membrane. So it has to go through channels, and one of the channels is located on ATP synthase, which powers it. So something that you should know for overview. But before we get into all the complexes as well, talking about electron acceptors, we know that we have NADH, FADH2. We use ubiquinone as well. We also use the cytochromes like cytochrome C. And the way that these are all transferred is through iron sulfur clusters, especially in complex 1 and 2. We also know that there's the copper atoms in cytochrome in the fourth complex. We know that there's a bunch of them, and you should memorize them for sure because Holmes may ask you, hey, what's the electron flow in complex 1? Or what's the total electron flow? So it's good to know all the specific electron acceptors and donors that are donating the electrons, accepting them, and everything. So also another thing to keep in mind is that these electron acceptors are usually very conjugated, meaning that they're either aromatic, in this case right here with ubiquinone. Ubiquinol, it forms a benzene group. We know benzene is aromatic. And sometimes it forms this conjugated pi system right here, where if we add a negative charge to it, or we take away that when we add that negative charge, it's going to be able to, by resonance, basically destabilize more. So that's why conjugated pi systems and aromaticity is important, is because we can stabilize that negative charge. And that's what makes them good electronic. Over the basics of ETC, we know that the only complexes that pump protons are complex 1, complex 3, and complex 4. And the number that you should commit to memory is 442. 4 protons in complex 1, 4 for complex 3, and then 2 for the last one. And complex 2 does not pump any. And these 4 complexes that form the ATC, you should know the names, but I'll explain them so it makes more intuitive sense. For complex 1, we have this NADH ubiquinone oxidoreductase. Now, it sounds complex, but literally it is telling you what is being reduced and what is being oxidized, right? So in this case here, NADH is being oxidized, right? Right here, we see that's being oxidized, the electrons are being pulled away. And our ubiquinone is going to be reduced. The one is the suffix for a ketone. So when it's formed the QH2, we call that ubiquinol. But this is just telling you your substrates that you're working with. And then oxidoreductase because it's an oxidation reduction reaction. That's how you memorize the name. And then for complex 2, we do know that succinate dehydrogenase is part of the Krebs cycle. So this one's like pretty intuitive. You just have to remember that, okay, succinate dehydrogenase is on the membrane itself, and it's going to participate in ETC as well as Krebs, like simultaneously. And then complex 3 is cytochrome BC1 complex. The reason why we call it cytochrome BC1 is because, one, it forms cytochrome C, so that's one way to memorize it. In terms of cytochrome B, I don't know what that's for, but we'll go into detail. But that's one that you have to memorize, and that protons are pumped with this complex. Complex 2 does not pump any. And then complex 4 is cytochrome oxidase. We're going to oxidate our cytochrome C. That's why it's called that. I'll get back to you in terms of the cytochrome B once we hit complex 3 more in depth. And we're creating this proton gradient every time we go through complex 1, 3, and 4. Complex 2 does not contribute to the proton gradient. So if you ever get a question of like true or false, does complex 2 contribute to the proton gradient? It does not. It is false. And then the end step is we are going to transfer all the electrons because the electrons are very high energy. And if they don't get transferred to the oxygen, where do they go? And if they stay in this complex 4, you're just going to basically ruin it because you're going to put so much high energy into it. It's going to destabilize the whole protein. And so our oxygen turns into our final electron acceptor, in which we take 2 protons, we add the 2 electrons, and then they combine with the oxygen here. And it's going to form our 1 water molecule, right? And we would double this, obviously, if we were trying to deal with whole number molecules. So one thing I want to emphasize, again, with the protons and complex 1, complex 2, so I'm going to walk through the route of NADH briefly and the route of FADH2. So for NADH, we see that the red rectangles represent NADH and the proton flow. So NADH basically gets oxidized at complex 1. The 2 electrons do their thing. I'm not even going to worry about that. We know that complex 1, it pumps 4 protons across. Then as the electrons get passed on over to complex 3, see this arrow, it's not saying that QH2 is going to complex 2 and then 3, it just passes 2 and goes straight to 3. Once it's at 3, it's going to, again, move the electrons to cytochrome C, and the 4 protons are going to be pumped again. From cytochrome C, then we go to complex 4, in which, again, the last 2 protons are pumped through. So something to keep in mind. And then for FADH2, we're going to entirely skip complex 1. We're not going to deal with this. So we're not pumping these 4 protons anymore. We're skipping that. And that's very crucial because we know the proton motive force is what drives ATP synthesis. So this is going to be a less efficient, is what you could say. And so FADH2 is going to come into complex 2. It's going to do its thing, not pump any protons, go straight to complex 3, and then 4 protons are going to be pumped. And then the last 2 in complex 4, once cytochrome C does its thing and transfers the electrons over. So we know that it's 4-2, so it's 6 rather than 10. So something to keep in mind, and definitely is going to help you with calculations later on. So moving into complex 1, overall function is just to transfer your electrons from NADH to QH2. So essentially, these arrows are backwards. It should be the other way around. But NADH2 is going to pass the electrons, and it's going to go through complex 1 and eventually be passed to ubiquinone, and it becomes ubiquinol. And while this happens, when these electrons are transferred onto ubiquinol, there we go. Tongue twister. It's going to do like this simultaneous pumping of protons across the membrane. And one of the things that you should memorize is the cofactors. So we have FMN, which is one of them, the first that received the electrons from NADH. And then we have the iron sulfur clusters that take those electrons from the FMN and coordinate the electron and direct it towards ubiquinone. Without these iron sulfur clusters, this complex would be not as efficient. It wouldn't be able to tell the electron go this direction. The electron will just go wherever it wants. It's high energy. It wants to lower its energy, right? If you have a free electron. And so the end result is that you reduce ubiquinone to ubiquinol. And moving on to complex 2, we have succinate dehydrogenase. So the overall function is to transfer the electrons from succinate because it has higher electronic energy than fumarate. It's going to transfer those electrons to FAD and then basically form FADH2. FADH2 is just going to pass it to iron sulfur cluster. So the FMN that we saw in complex 1 is no longer present here. So the FAD will then pass on to iron sulfur clusters, and then it'll pass it on to ubiquinone to form ubiquinol. And so again, with this complex, no protons are pumped across the membrane. And then this bypasses complex 1, as we mentioned before, so I won't even reiterate that. And this is a typo. This should say similar principle to complex 1, but that's the same thing. And then this is the same enzyme for TCA step 6. So from succinate to fumarate, we know that it's succinate dehydrogenase for that, and so it's the same exact enzyme. So moving on to complex 3, the overall function is to transfer electrons from our now reduced ubiquinol, QH2, to cytochrome C. And so the electron flow is going to go from ubiquinol to iron sulfur clusters to heme C1 and cytochrome C. And I think the reason why we call it cytochrome BC1 is because there's cytochrome or heme Bs that also help with the Q cycle. So I think that's why we call it that. But again, with this complex 3, we're going to pump 4 protons across the membrane per ubiquinol that we reduce, or that we oxidize, sorry. And so the 2 QH2 molecules transfer 2 electrons each, and so it's 4 total. And so we regenerate 1 QH2 molecule at the end. So the net result, we're going to, and we'll explain the Q cycle. I'm just telling you this now because it's right on the slides, is that there's going to be net 1 QH2 used because we end up producing 1 in the end. So even though like 2 go in, we still produce 1, so 2 minus 1 is 1. So we end up just using 1. And then 4 protons are pumped per QH2 molecule. That's why we call it that. So we'll go into the Q cycle now, and I'll explain this much more slowly. I know I've been talking fast. That's because the other stuff is very brief overview. So what we're doing with the Q cycle, again, remember, we're in complex 3. We are trying to transfer electrons from a 2 electron carrier to a 1 electron carrier. So we know cytochrome C here, it only can only hold 1 electron. It cannot hold 2. So if we have run into an issue of this holding 2 and this can only hold 1. So the issue is that 1 electron is going to go to cytochrome C. No issue. That's good. But what about the other electron? That's the issue that we run into. And this is where the Q cycle comes in. So our heme Bs, which again are not part of the electron flow from the QH2 to the sulfur cluster to form cytochrome C, the heme Bs are not a part of this electron flow going upward. Heme Bs are the thing that are keeping the Q cycle going, right? And so with these electrons right here, they're going to pass on these, and you don't need to know the L or the H. It could just be low and high, literally. I don't know. But these electrons are going to get passed on to a free ubiquinone, and it's going to form a radical, essentially. So again, when there is the first QH2, right, when it comes in as well, as these are past ubiquinone and you form this radical, 2 protons are going to be pumped, right, with 1 QH2. But remember, we form this radical and we don't want our ubiquinone to be in a radical. It's not going to want to stay as a radical. So what we have to do is introduce a QH2 for another one, a second one. So the second QH2, imagine that it just came in, right? And now we have the 1 electron going to cytochrome C, no issue. That this upper part makes sense. What's confusing is this bottom. What ends up happening is the second electron from the second QH2 is going to go through the hemes, and then instead of just this free ubiquinone, our ubiquinone radical is just going to stay here, right? And that's where it's going to grab that last electron to form QH2, and 2 protons are going to be grabbed from the matrix to form our now newly regenerated QH2, right? And so we also, again, pump 2 protons again. That's why we say for one right here in this back slide, we say that 4 protons are pumped across the membrane per QH2. And that's because, again, we introduce 1 in the reactants and another one. So that's 2 QH2s in the reactants, right, on the left. And then we regenerate 1 on the right. I don't know why there's balloons. But essentially, what ends up happening is for the net reaction, we just do 2 minus 1, which forms 1 at the end. And so the end result is that we're technically only using 1 QH2 because we regenerate 1 in the product. So hopefully that makes sense and it's not confusing. Here is the reactions, like their respective reactions, like steps from the Q cycle that are listed. The only thing that I just want to suggest or emphasize again is that this QH2 in the reactants, this is for the first Q cycle, right? And this is for the second. We have 2, but we also regenerate 1. So it's almost like a canceling reaction. And so we really only use 1. And yeah, this is just another way to say it. It's a good thing to review on your own time if that was a little bit confusing. So for complex 4, cytochrome oxidase, right? The overall function is to transfer 4 electrons from 4 cytochrome C molecules to O2. So what this means is that we're going to have 4 separate cytochrome C molecules come in. They're going to bring their electrons. They're going to get transferred from the cytochrome C to the first copper A, and the copper A is going to transfer it to these iron copper cluster centers. And what's going to end up happening is it's going to transfer it to heme A, and then heme A3, and then copper. So one thing I want to emphasize is that with the electron flow, we go from cytochrome C, CuA, iron copper clusters, the heme A, the copper B and then the oxygen. Once the electrons reach this copper B, the oxygen comes in and will basically accept those electrons. And when that happens, simultaneously 4 protons are pumped, along with 4 of the protons being substrate. So think of it as like 8 in total, but 4 of them are realistically contributing to the proton motive force. And this is what I want to emphasize. Each pair of electrons donated leads to 2 H pluses that are pumped, right? So each pair of electrons, so we have 4, so each pair, so a pair is 2, so 4, so there's 2 pairs here. There's 2 pairs. That leads to 4 protons if we have those 2 pairs. That's why we end up with this 4. And so again, 4 protons are used to make water because each O2 accepts 4 electrons. And so this is the half reaction or reaction, whatever you want to call it, that forms our waters at the very end. And so here is a review of all the complexes. I recommend going through an animation slide, like after you finish watching this entire thing and taking your time through it, but I'll just skip through it. Definitely something to look at later on and test yourself. And then here's a practice question on it. I will let you guys go through this. The answers are in the answer notes. So now moving on to ATP synthesis. We did the 4 complexes. What do we do after? So we have this chemiosmotic model of ETC, which all it means is chemiosmotic, which just means the chemical concentration gradient that drives ATP synthesis. That's literally it. And it says here, ATP synthesis is driven by a proton motive force. The protons are building up in the inner membrane space, and they want to go into the matrix. They really, really want to. So ATP synthase utilizes that and says, hey, we have a channel. You go through this, you're happy, and you get to go down your concentration gradient. But we're going to take the energy. That's all it is. That's really what ATP synthase, how it works and how it's powered. And so complex 1 through 4 creates this ETC proton gradient, except complex 2. But I mean, theoretically, you could say that later on downstream it does contribute. So I won't get into the very specifics. You know exactly which complexes will transfer the protons through. So I want to emphasize this point. The inner membrane space, this space here, it has a low pH. Remember that low pHs correspond to high proton concentrations. So whenever you see H concentrations being high, not necessarily positive charge, because I know that gets really confusing, but whenever the proton concentrations are high, you can assume low pH. You can assume acidic. So that's something I want to emphasize. And because the protons are leaving the matrix itself will have a lower concentration of protons once it's pumped. So that means that the matrix is higher pH, and it's going to be more negatively concentrated. And I think that's 8. And then last point I want to say is that ATP synthase rotates because of the electrochemical potential, and H plus moves down its gradient, while ADP will convert into ATP. That's literally it. So let's have a closer look at ATP synthase. ATP synthase has 2 parts. It has a F0, which is this golden part right here, and it's essentially our thing that locks it's like our rotor, like an engine, right? Think of it like the engine. If you have a car without an engine, it's not going to drive. In the same way that to form ATP, we need an engine there in the first place. And so you can think of like the protons as the gas. This is just an analogy. If it doesn't work, don't worry. Ignore it. So then the F1 subunit is going to be your gamma rotor, this green part, the epsilon, which it's not even that important, so don't even worry, and then the alpha beta subunits. And one thing I really, really want to emphasize is that the beta subunits, these purple ones, are the ones that catalyze this reaction right here from ADP to ATP. Alpha subunits do not do that. So I want to talk about the energetics really quick, just to touch a brief overview on it. So this is just a typical reaction right here. I'm not going to go into it. The ATP synthase itself is an enzyme. So you would think, oh, doesn't it follow this curve right here? Well, no, because remember, Michaelis-Menten assumes that we have our free products, or free substrate and free enzyme, and then they combine to form the complex, right? And then it conducts from ES to EP. Right? And it's still in the complex. It's not freely dissociated. And what this diagram is telling you is that the highest activation energy is simply just the ATP dissociating from the enzyme. So it's this that's really the activation, or the highest activation energy, the rate limiting step. It's not this conversion. It's not this catalysis it's not the phosphorylation of ADP into ATP. So again, that's why this first point is true. F1 binds to ATP with extremely high affinity. As you can see here, it is extremely stable. It's very low, like the delta G is very stable. It doesn't want to leave this spot. In order for the ATP to dissociate, it has to gain a lot of energy for that dissociation. And what's the thing that causes this activation energy to be fulfilled, to like occur so that we actually move towards the right? It's the fact that we have that proton gradient being coupled to that rotor, that gamma subunit. And like I said, this is the channel. It powers that rotation of the gamma subunit, and it forces the ATP out. That's how this works. And so if you isolate the F1, meaning that you get rid of this F0, pretend that this yellow part is gone. What will end up happening is that our ATP synthase is actually going to catalyze hydrolysis, and it occurs in an equilibrium. It doesn't go just one direction, right? It'll catalyze towards the ADP and inorganic phosphate direction. And so this is where we use an IF1 inhibitor to counteract, meaning that an inhibitor will bind to like the beta subunit and stop the catalysis from happening, which is one of your experiments, but we'll talk about this in depth. I don't want to jump the gun yet. So there are 3 beta subunits that can bind ATP and ADP and exist in 3 different states. We'll go more into this with a clear picture on the next slide. I just want to emphasize that one site has ATP, one site has ADP, and one site has nothing. Right, you're always constantly interchanging, right? This one has ATP, but then once the gamma subunit rotates and gets rid of this ATP, right? Right here, basically the gamma subunit is in the beta empty. There are 3 beta subunits that combine ATP and ADP and exist in 3 different states. And we'll go into depth on the next slide. Essentially, we have ATP in one spot, we have ADP in another, and then we have an empty. And the reason why it constantly interchanges simultaneously is because we want efficiency. We don't want to find ourselves with 2 empty sites and 1 ATP forming from it, right? We want to constantly be generating ATP because if we need to work overtime, like let's say we're sprinting, for example, and the muscles need ATP right away, we don't want to be stuck with 2 empty beta subunits, because then imagine this happened on a scale of hundreds of thousands of ATP synthesis. We would not be producing that much ATP for the muscles that require it, So it constantly has to be rotating and alternating between these 3 different states constantly. You will never find a ATP synthase that has 2 empty beta subunits or all of them are ATPs. You will never find that. You will always find this interchanging. And so we'll talk about this on the next slide, how this works mechanistically. So this is moving in a counterclockwise direction. And all I want to say is let's start on the top. That's all we need to know. So we see that the gamma subunit almost like occupies that space, that empty space, this empty beta subunit, and that's actually what forces the ATP out. So let's say the ATP has already been gone, right? We're talking about this unit, and the gamma subunit is inside here, right? Well, what's going to end up happening is once 3 protons are pumped through the channel, it's going to power that rotor that we talked about, that yellow portion at the bottom, and what it'll do is it'll literally force the gamma subunit to go in here, right? And it'll force the gamma subunit to go into this direction where there's already an ATP formed. And that is exactly what's going to power this dissociation. Again, this is our highest activation energy. That gamma subunit is going to force the ATP out. So that's where we make up this high activation energy, is the fact that we're forcing the gamma subunit to turn into it, and it's powered by the proton motive force. That's how this whole thing works. And again, when that occurs, ADP plus the inorganic phosphate is going to react as well, because they're all mechanically coupled. These 3 sites will react simultaneously. ATP gets kicked out, these are forced together, and then a fresh ADP and inorganic phosphate is going to take the empty beta subunit space here. And so that's exactly what happens. It occurs all simultaneously. And that's where we really need to emphasize, is that if we were to stop one of these subunits and it wasn't able to move anymore, the rest of them would not work as well. They're all mechanically coupled. That is the key word. They are mechanically coupled. And so for every 120 degrees, the gamma subunit comes in contact with ATP containing beta, and it kicks out the ATP, which is simply what we just said here. When it goes from this beta subunit to this one, it's going to basically force it out. And then efficiency is key, as I mentioned before. Don't need to go over that. And I want to emphasize this. We need 3 protons to flow down the gradient back into the matrix. That's what we need to power the rotor. That's exactly the energy that it takes to form our 1 ATP. So something to keep in mind. And then adenine nucleotide and phosphate translocases. So you may say, well, why is this slide important, right? Well, remember that I said that a fresh ADP and inorganic phosphate is going to come in. And this is on the matrix side. Remember, the ATP synthase, right here, I'll show you on this diagram, is in the matrix side. So we need to have a constant supply of the ADP and inorganic phosphate coming into the matrix, or else we're going to run out. And then what do? Then we run into the issue of having 2 empty beta subunit sites, or all 3 are empty, and then what do? If we need the ATP, we need to regenerate that. And that's exactly what these phosphate translocases are. So how the matrix gets these substrates is through these two translocators, antiporters and symporters. The first one is adenine nucleotide antiporter, and I don't call them translocase, I call them antiporters or symporters just for simplicity's sake. So the first thing is we're going to have ATP, ADP, and the ADP is going to pass from the inner membrane space into the matrix. Again, we want to never constantly run out of this supply. And what ends up happening is the ADP will combine with the inorganic phosphate, and we'll go over that in a little bit, and it's going to form our ATP. ATP then leaves because we need to use it in the cytosol. We need it for these proteins that are for PK, like protein kinase A, protein kinase C, a bunch of these proteins that are in the cytosol, right? We need to harness that ATP for it. It needs to leave the mitochondria, and that's what this antiporter is used for. So now, moving on to the phosphate symporter now. This is where we're going to get our inorganic phosphate, and I know this isn't showing the PI, but that's exactly what it is. It's just protonated. It's phosphoric acid. And this is a symporter where we have the phosphoric acid, the H2PO4, and it's a minus, plus the proton, and it's going to come in together and go into the matrix from the inner membrane space. And that's what supplies our inorganic phosphate. And these two now beautifully come together and constantly keep the supply of ADP and inorganic phosphate to form our ATP. And before I move on, I just remember this, is that we're talking about electrogenic transporters and electroneutral transporters. So the ATP ADP antiporter, this adenine nucleotide antiporter, is a electrogenic transporter. What does that mean? Well, take a look at the charges. The ADP has a 3 minus, the ATP has a 4 minus, right? So what ends up happening is a -3 charge comes in and a -4 comes out. What is the net? The net is that a negative charge will leave. And so that's exactly what electrogenic means, is that your charge in the membrane potential between the matrix and the inner membrane space, not to confuse topics, but the charge is going to basically cross the membrane and it's going to equilibrate, if that makes sense, because our negative is going from the negative to the positive. So it's going to equilibrate. That's what we call electrogenic. Whereas this one, the phosphate symporter, is going to be a electroneutral transporter. And what does this mean? Is that we have our negative here, but we also have a positive coming in. And so those cancel out to be 0, and that's why it's electroneutral. So moving on. So let's talk about uncouplers of ATP synthesis. So we know that ATP synthase is not 100% efficient, and there is actually a evolutionary reason why. So ATP is only about 38% efficient, and the rest of the energy is lost as heat. And we can generate more heat by partially dissipating the gradient without making ATP. And how can we do that? Is through uncoupling proteins, which one example here is thermogenin, and they're usually called UCPs. And what these do is they essentially open, and the protons are able to flow down their concentration gradient. And what ends up happening is we generate heat. And with this heat, I will say, from an evolutionary perspective, something so that you can understand why it's not 100% efficient, is because as mammals, we need to have a internal body temperature of 98.6 degrees Fahrenheit. I think that's the number. Don't quote me on it. But we need to be extremely warm, right? We need that heat. So we need sometimes to uncouple if we're cold or if to maintain our internal body temperature. That's why we have this uncoupling sometimes. And this is exactly why in brown fat, when brown fat is most prevalent in infants, in babies, in newborns. And that's because they need to stay warm as they develop. You don't want to expose them to cold environments because that induces shock, all this physiological changes from prolonged cold exposure. So the heat is going to keep them warm because the brown fat tissue has more prevalence of these thermogenin uncoupling proteins, and that's what generates more heat. And so when we're talking about white adipose tissue, for example, that we see in adults, those ones do not generate heat. And yes, they generate ATP synthesis, but they don't have as much prevalence of this UCP1. So that's why we have this in terms of mammals. When you talk about cold-blooded, I have no clue. I don't know. But just wanted to give you some rationale as to why we have these things as well. So here's a question about brown fat. You can look at the answers down there. And then now talking about shuttles. So we've addressed so far ETC through all the complexes, complex 1 through 4, and ATP synthesis, or ATP synthase. Remember in step 6 in glycolysis, we produce our NADH, right? We're basically converting the glyceraldehyde 3 phosphate into 1,3-bisphosphoglycerate using glyceraldehyde 3 dehydrogenase. And so you may say, well, why are you bringing this up, Alex? Well, this is the whole point of the malate aspartate shuttle, is that these NADHs are in the cytosol. They are not inside the mitochondrial matrix. And what we need when we need them to do is to translocate inside. And that's what this shuttle is used for. We're basically going to utilize this malate dehydrogenase, this Krebs cycle enzyme. Remember, this is the Krebs cycle right here, this reaction. And this enzyme is also found in the cytosol, in this intermembrane space. These are basically like synonymous. I don't even mention that. But point is that our oxaloacetate, remember that malate dehydrogenase is reversible as well. And oxaloacetate can very much become malate and basically be reduced and take in the energy from this coenzyme, right, the NADH. And so the malate is now reduced, and it's carrying the proton, the electrons from NADH, and it'll transfer into the mitochondrial matrix using this malate alpha ketoglutarate transporter. The reason why it's named that is because it's an antiporter. There's a malate going into the matrix and a alpha ketoglutarate going out, right? And so once the malate is inside, what's going to end up happening is this malate dehydrogenase is going to work again. And now we're going through Krebs cycle, and we're going to harness the NADHs. Now that they're on the proper side, they can actually work with complex 1 and start producing ATP that generate that proton motive force. They can do all that. And I just want to go over this bottom stuff because it also is important too to know, but not as important as this top part. But essentially, we're going to have our malate go into oxaloacetate, right, after it donates the NADH, or after it donates the electrons to NAD plus to form NADH. The oxaloacetate is going to basically do this aminotransferase reaction, meaning that we're transferring an amine from glutamate onto oxaloacetate. And what ends up happening is we form an aspartate. And this is very easy to memorize because it's a malate aspartate shuttle. These are the two substrates, the two molecules that can go across the intermembrane space the inner mitochondrial membrane and go from matrix to cytosol or cytosol to matrix. I know this is intermembrane space, but they're synonymous because the outer is permeable. And so here the aspartate is going to also, it's an antiporter as well. These are all antiporters. And the aspartate goes out and our glutamate comes in, sorry. And remember, in this reaction, we also form aspartate, but we also form alpha ketoglutarate, which alpha ketoglutarate is just the carboxylic acid of glutamate. That's a very good relationship to know, especially coming up in lecture, I think 17 or something like that, 16. I don't want to make it a stressing point, but you will need to know like the carboxylic acid derivatives of our amino acids. That'll be later on. I don't want to stress you out. But I think that's everything I needed to cover on this slide. Yeah, let's move on. So there is another shuttle as well, because let's say, for example, right, this is a scenario that you could see this being prevalent in, and it could be tested in an exam. So you can thank me for this example. But essentially, let's just say that we're sprinting outside right now, and we're sprinting really far, and it's going to be a while, and we need to utilize our ATP stores, right? And we're going to obviously, we know sprinting is going to favor anaerobic respiration. Why is that? Well, that is because, again, glycolysis will produce our NADHs, and the NADHs that form in the cytosol, because glycolysis is promoted in anaerobic respiration, is that our NADHs are going to react with the cytosolic glycerol 3-phosphate dehydrogenase. And what ends up happening is it's going to reduce our dihydroxyacetone phosphate. Again, it's an acetone, so it's a ketone, and it's going to turn into an alcohol, glycerol 3-phosphate. And this glycerol 3-phosphate is going to interact with the mitochondrial glycerol 3-phosphate dehydrogenase, which is on the intermembrane side. Now, this is important because it automatically becomes from FAD to FADH2, and it'll go basically straight to ubiquinone, and it'll go into complex 3. Now, this is important. I bring up the muscle example because we need to harness the ATP fast. We need it, and we're not going to go through Krebs cycle. We're not going to go pyruvate dehydrogenase. We're not going to take that long route because we need it fast. There's a temporal need for it, and so this is what the shuttle is for. And remember, this is bypassing complex 1. Remember that if we bypass complex 1, we're no longer pumping 10 protons, we're pumping 6. And what ends up happening is that since we pump 6 protons, we're going to pump less protons across the ATP synthase channel. And that means we generate less ATPs. And so that's a bad thing. So for long-term use, this is not the favorable pathway. This is not a favorable shuttle. The other one is because we form our NADH, which goes through complex 1. That's the difference between the two, is that this one is extremely efficient, this one is not. Because again, you can treat this similar to complex 2 in that it doesn't pump protons and it basically goes straight to complex 3. You can treat it similar, but it is not. This mitochondrial glycerol 3-phosphate dehydrogenase is not succinate dehydrogenase. And I want to talk briefly about energy charge of a cell, which all it is telling you the adenylate forms that can be used as energy sources. So that's why this is half, because half the ADPs can be formed into AMP, which means that they're used as energy, or the other half can be forming ATP. And it's just taking the number that you can use as energy, and then out of the total number of nucleotides in the cell. And you don't need to know the specific stuff. All you need to know is that a cell likes to maintain a energy charge of 0.9. That's pretty much it. And the number can only be from 0 to 1, and that's because you can never exceed the total concentration. You can reason that yourself. And this is another question for you for cyanide, sorry. This is a nice diagram of all of the glycolysis, Krebs cycle, etc., how these energy molecules can affect it. Some of them are indicators of low energy, so they activate, cellular respiration. We'll call all these metabolic processes cellular respiration. The low energy activate these, and then the high energy say, hey, hold up, we already have too much energy, we don't need to keep going. And so that's what they do. That's how you can think of this. And some of these molecules, yeah, you're right, like citrate is not necessarily AMP, ATP, or NADH, but it is a signal of, hey, we have too much citrate, our Krebs cycle is working at optimal, and we need to slow down on glycolysis in order to use up all the substrate. So that's all it's basically acting as. And this is just text form of that previous diagram. If you want to look through this, I'm just going to breeze through it, like not even say anything. So you can look at that. And then this is just a nice, neat summary. The thing I recommend that you solidify is these numbers. 1 NADH of the matrix forms 2.5 ATP. 1 FADH2, whether it's in the cytosol or whether it's in the matrix, will form 1.5. And then this malate aspartate shuttle and also the glycerol 3-phosphate shuttle, they're different in that the malate aspartate, remember, it conserves it because we move our NADH inside. We go from the outside, the cytosol, into the matrix. Whereas our glycerol 3-phosphate, we don't care about making the NADH on the inside. We just need the energy right now. And so that's exactly what this is going to do right here is we're going to bypass the NADH and form FADH2. So that's the difference between these, and that's why this one we get 2.5 because it goes through complex 1. This one with the glycerol 3-phosphate shuttle is 1.5 because it only goes through complex 3 and 4. And we skip complex 1. And again, each mole of glucose can create 30 to 32 ATP depending on if the shuttle is G3P or if it's malate aspartate, and you can run through the math yourself. And this is saying is that the energy is 32 to 34% efficient, which is really high. And a real world example is that engines are only about 30% efficient. Something cool to know. All right, 40 minutes, bro. I feel like I've only been talking for like 10. Anyways, moving on to the next one. Let's do this. Lecture 11, photosynthesis and carbon fixation. We're gonna go through this. I'm gonna break this lecture down because I feel like it could be presented better. But point is that let's give an overview about redox chemistry. And all I wanted to say is that given this half reaction right here, the standard reduction potential, when we're basically reducing the oxygen, as you can see, we're adding 2 electrons to oxygen and the 2 protons to form water. This is the highest electron reduction potential. What is that? What am I trying to say? Is that if we just let everything spontaneously happen, no more enzymes, no more photosynthesis, everything would become water. But we know, given that we're breathing oxygen, that we're still creating oxygen. We're not running out. So what is exactly going the opposite direction? That's all this slide is just trying to say. And the same thing with carbon. I know carbon isn't shown here, but we always talk about breaking down carbons and forming carbon dioxide in cellular respiration. How do we go from carbon dioxide to glucose again? How? So that's what plants do, and that's what we're going to go over. So talking about photophosphorylation. Phosphorylation, meaning we're forming ATP, and then through photosynthesis, through these light reactions, through light. And so this is an overview, is that the sun's going to come in into the chloroplast, and we're going to have these light reactions occur in the thylakoids, and that's going to form our high energy molecules. So we haven't formed the glucose yet. This is just saying, hey, we got the power to make glucose now. And so that's what the light reactions are doing, is that we're saying, the sun, thank you for giving us all the energy. We're going to use that and create these high energy electron carriers that are going to be able, or one electron carrier, and then also high energy molecules that are going to be able to create these very long-term storage carbohydrates. That's all it is. And the carbohydrates are assembled through the Calvin cycle or the carbon assimilation reactions. And I will talk about water being as a source of electrons much more in depth, but that is something you need to know, is that photosynthesis, it produces oxygen. So where does the oxygen come from? It comes from the water. That's the way I usually think about it and memorize it. But we'll talk about exactly why this is extremely important. So Don't worry. So talking a more brief overview about the chloroplast, they have 3 membranes, not 2 like the mitochondria. So there's a porous outer membrane, meaning that it's impermeable to the inner membrane. There are 3 layers. There's the porous outer membrane, there is the impermeable inner membrane. So this outer membrane, the stuff can go in and out. The inner membrane, stuff cannot go in and out. You're going to need that extra like shuttles, the channels, and all that and transporters to pass molecules through that are bigger and that cannot diffuse freely. So inside the inner membrane, outside of these pancakes and inside the inner membrane, it's like this green space. I know I can't highlight it that well, is the stroma. And then the stroma contains thylakoids, these pancakes. And inside the pancakes is considered the thylakoid lumen. And that's our third layer. That's our third membrane, is the thylakoids themselves have the thylakoid membrane, is our third layer, and it's what separates the lumen, the inside of these pancakes, from the stroma. And there are 2 processes. The light reactions occur inside the thylakoids. That is where our chlorophylls are found. And then the carbon assimilation Calvin cycle, where we form the sugars, are in the stroma. So talking about the light reactions, don't worry about the Calvin cycle. We will go over that in a little bit. So for the light reactions, we are talking about inside the thylakoids. So keep that in mind. We're inside the thylakoids now. So the pigments are basically these conjugated pi bonds. So if you ever see, conjugated pi bonds, think these things can carry high energy. Therefore, they're like chlorophylls. They're pigments. They can harvest light energy because they can take that exciton, we'll talk about this in a little bit, and relay it onto other nearby chlorophylls, pigment molecules to reach the special pair. And we'll talk all about this in a little bit. I just wanted to emphasize pigments. And there are 2 types of things that can happen with a pigment, right? We can have an exciton that can be transferred to neighboring pigments, which I just talked about, or they could just simply go back down to their ground state and fluoresce, show that color. And that's exactly what we see here, is that sometimes there's a certain wavelength that will basically be reflected or absorbed. And that absorption means that these electrons will be excited, and when they rest back down, that's when they're going to reflect that color. That's when they're going to fluoresce and show, like for example in chlorophyll B, the green. So now talking about this very confusing diagram, I will break it down because I had a hard time understanding this the first time. So what we see here is light's going to come in, right? And we have these antenna molecules. These are not our special pair. These are just molecules that are chlorophylls. They're pigments. They can absorb light. And the light's going to come in. It's going to excite this electron inside the antenna molecule. And it's going to excite it. Once it's excited, nearby antenna molecules will also want to grab that electron, right? And so that's exactly what happens, is the excited electron from the first antenna molecule will go to the second. And this is what we see here. And then the same thing is going to happen from that second antenna molecule to the reaction center, this special pair in a way. Now, here's what I want to emphasize. The special pair is poorly illustrated. It's telling you right here that, and what it's insinuating based on this is that, this is middle energy level, this is high energy, and this is low. That is not what it is. These should technically be degenerate, almost the same level. And what I'm trying to say with that is that when the excited electron from the antenna gets passed on to this middle one, this special pair, this reaction center, it's going to get excited, right? And an electron acceptor, some random molecule, it's not higher energy, just some random molecule is going to be hey, I'll take that from you, and then take it. And that's where the electron goes. That's what this is saying. It's not going higher in energy. It's just the fact that the electron acceptor is nearby and will grab it. And it's coordinated to be so. Right? The special pair is special in that the electron acceptor is right there and the electron donor is right there. So that's what this graph is trying to insinuate, but I got the idea that, it just jumps higher energy, but that's not true. How is it going to get higher energy if there's no extra light being added? That just doesn't make sense. So these are technically the same energy level. It's just that they're showing it spatially they're next to each other. And so going back a little bit, digressing, we have this electron acceptor that just grabbed the electron from that middle special pair, right? So the electron acceptor is going to now go do its thing, go pass on the electrons, do whatever, go do photosynthesis. And then the electron donor, that's another molecule that's nearby, and it's going to give its electron up because we have to restore the electron here. It doesn't just magically come back. It has to be restored from somewhere, and that's what the electron donor is for. And then and then at the very end, we have an empty electron donor, but a new one comes in. And so this is not necessarily has to be refilled, new one comes in. And I'll explain this in the context of photosynthesis that we know. So let's get this And just before I go into it, just one thing about the reaction centers is that there's this special pair, and all it is, and this is the only thing I really want to emphasize, don't worry about this QA, QB, don't worry about that. The special pair, all it is that it transfers energy from energy faster to neighboring pigments. It's all like coordinated. And that's what I was trying to tell you, is that the electron acceptor, the special pair, and the electron donor are all like coordinated with each other so that there's a very fast transfer. That's all it is. And so I wanted to orientate us to this battery diagram, but with actual labels here. So as you can see here, our reaction center, and we're talking about in photosystem 2. In this diagram right here. So I'm going to constantly go back and forth from it. I'm not going to even bother reading this text because I think this is important to first discuss. So with P680, right, it's photosystem 2. What ends up happening is our 10 molecules will pass on the light, and eventually it's going to find its way to P680. Now, what ends up happening is the P680 gets excited. This electron here is going to get excited, like in this diagram back here. We see that it goes from the ground state to the excited. And that's what we see here. It goes from the ground state to the excited, the star. Now, our electron acceptor is going to be the pheophyton, because that's exactly where the electron gets donated to. Right here, it's going to get excited and then pass on to pheophyton. It's not necessarily that it's higher energy. It's just the fact that it just gets passed on. And if anything, pheophyton is a lower energy, so the electron's going to want to pass and not go back, so it can't go in the back direction. So that's one thing to note. And then the electron donor in this case is going to be our water. This is what I was talking about. Water is necessary. It is our electron donor because we're going to harness these electrons using this oxygen O2 or oxygen evolving complex, and it's going to take the electrons and pass them on and donate them to photosystem 2 to regenerate that electron that should be gone essentially after it does this first excitation, right? You can see here the electron is not there, so the electron donor is there to pass the electron on. And that's exactly what that is. So hopefully that breaks down that diagram. It is not the fact that, I don't know why I understood it, like why I thought of it like this the first time, was that, oh, it gets excited even more to get passed on. That is not what happens. It's just transferred. That's it. Okay, so now we'll talk about the actual like plants, like Z-scheme, all this stuff. So each electron that we see that goes through this entire thing is excited twice. Photosystem 2 at this P680, and then photosystem 1 at P700, where the light is. And light is essentially exciting photosystem 2, and it basically will excite it. It gets passed to pheophyton and then plastoquinone, which is what, it's very similar to this, but I just didn't want to confuse you guys with this QA, QB, so you can just honestly ignore that. But it gets passed on to plastoquinone, QA, and then that gets passed on to QB. These are different like isoforms, so you don't really need to know specifics. But the plastoquinone is going to transfer the electrons to cytochrome B6F complex. And this is a special complex that can pump protons, and also it can basically react with the ferredoxin along with the plastoquinone. And after this cytochrome B6F, once it's excited, it's going to transfer that electron onto plastocyanin. And plastocyanin, now if we were to take this battery analogy and do it for photosystem 1, the middle would be, what would it be? It would be P700. That's what the middle would be, right? It's our special pair. And our electron donor is going to be the plastocyanin, because that's where the electrons are coming from to replenish P700. And then our electron acceptor, once the P700 is excited, is going to be our A0, A1. You can just think of it as iron sulfur clusters, and then our ferredoxin is the ultimate fate, right? So that would be the electron acceptor. And we'll go into detail about this. Don't worry if that flew over you. I have pictures of it in the next few slides. And one thing I wanted to know is that ferredoxin, or not iron is Fe. Ferredoxin can do a couple of things. It can either go a cyclic pathway, in which it'll literally go back to cytochrome B6F complex and redo the cycle again, and it's cyclic. Or, and the only reason why I would go this direction is to pump protons and contribute to the ATP synthesis. That's what it would do. Whereas iron could also go the non-cyclic route, in which it goes to iron NADP plus oxidoreductase. Now this enzyme, it's very complicated sounding, but like I told you with complex 1 and ubiquinone oxidoreductase, this is telling you the substrates that you're working with. The ferredoxin is your substrate. We know it has the electrons, and your NADP plus is your electron deficient substrate, meaning that this is going to grab it, and that's exactly what we see here. So that's what the non-cyclic is. And the ferredoxin has a balance between these two pathways. So sometimes you have too much ATP and you want to form NADPH. So the ferredoxin will prioritize the non-cyclic pathway. So there's a balance as well. And so going back to photosystem 2, talking about this manganese, I know it's very confusing. All you need to know is that manganese can hold 4 different charges: +4, +3, +2, +1, and then 0. Technically 5. It basically goes from a +4 oxidation state. Now, this is important to explain why we need 2 water molecules right here. We know each water molecule will hold 2 electrons, as given by this half reaction at the very, very beginning. 1 water molecule can hold 2 electrons, right? And we know that we can't have half an oxygen molecule. That just doesn't work. So we have to double everything. So that's why the manganese can hold a +4 state, is because we have to double this in order to form our 1 oxygen. So manganese can go from a +4 to 0 because of this, because of the 2 waters reacting with it to form our 4 electrons, which then reduce it. So 4 plus -4 is going to be 0. So that's why we form them in 0. Sorry if that was a little bit confusing. But basically, what ends up happening is we're going to send the electrons to the thylakoid membrane via our plastoquinone. That's going to basically go. Plastoquinone acts very similarly to quinone, as in it'll stay in the thylakoid membrane. You know, it won't like go into the matrix or in the cytosol or anything like that. Plastoquinone will remain in the thylakoid membrane, and it'll go straight to photosystem 1. So this needs more energy to generate than photosystem 1, and therefore that's why it's a shorter wavelength. That's why we see photosystem 680, right? P680, not P700. And we know that, just a little physics brief overview, wavelength and energy are inversely related. So shorter wavelength, higher energy. Longer wavelength, shorter energy. So they're inversely related. So just a little physics brief. So talk about cytochrome B6F complex. So what we're doing here is we're transferring electrons from plastoquinone, a 2-electron carrier, to plastocyanin. Sounds very similar to ETC, where we have quinone going to cytochrome C, ubiquinone, to cytochrome C. And in this case, again, we have the Q cycle. Hopefully that's still good. I'm not going to like really go that much into it. I don't know if it's on the next slide. Yeah, no. I won't go too much into it, but Q cycle leads to the proton gradient, as we've discussed. And this is where we see proton pumping. So cytochrome B6F does pump protons. And so that's something to keep in mind always, is that if you go through this, you will pass 4 protons through per pair of electrons, because the Q cycle occurs twice. And we know that the Q cycle occurs here, so it leads to proton gradients, and the protons are pumped into the thylakoids, which means that there are 4 protons being pumped every single time per pair of electrons. And the gradients are used to power ATP synthesis, which you guys already know. And ATP is needed for carbon assimilation reactions and to also for the regeneration reaction. So it's steps 2 and 3 of the Calvin cycle. We'll go into that in a little bit. So photosystem 1, right? The goal is to transfer electrons to ferredoxin from our P700, right? We're going to grab the electrons from plastocyanin, and that's going to donate the electrons, and it's going to get excited by light. And then what's going to end up happening is it's going to pass through these iron sulfur clusters and then pass on to ferredoxin. And the ferredoxin, if it goes the non-cyclic route, it'll form our NADPH, which provides the reducing power to do the carbon reduction step 2. And I think fixation as well. I don't want to speak too soon about that. And the cyclic path, it can either also go back to cyclic to contribute more to the proton gradient. And now I want to talk about like the spatial biology of photosystem 2 and photosystem 1. So photosystem 2, or reminder, photosystem 1, we know that it contributes to the non-cyclic pathway, meaning that it's going to help contribute to NADPH formation. Now, NADPH is a pretty big coenzyme, and we're going to need this photosystem 1 to be in a non-oppressed portion, meaning that there's no pancake stacking above it, it's not constrained, and so that the NADP plus can come in here and bind and do the reduction thing. That's the whole reason why it's in this non-oppressed. Whereas photosystem 2, all it does is just transfer electrons, and it transfers it to plastoquinone. Plastoquinone is in the thylakoid lumen. So it doesn't matter if it's in this in this oppressed area, right? If it's constrained by other nearby proteins that are folded in it on itself, right? It doesn't matter because it's just transferring electrons to the thylakoid membrane. So, and then one thing I want to talk about is, so that's spatially why they're located here, but also during midday, photosystem 2 activation outpaces photosystem 1. Why is that? Well, in midday, doctors will often tell you, hey, wear sunscreen. Why should you wear sunscreen midday around 3 p.m.? Because there's a lot of UV rays. Well, what is close to UV, or what is UV light in relation to visible light in terms of wavelength? Well, UV light has shorter wavelength. So UV light, if there's more prevalence of it, if there's a higher fraction of it, whatever you want to call it, then photosystem 2 is going to be excited more frequently than photosystem 1. Remember, it has to be a perfect match of the wavelength in order for it to be excited. And so And that's exactly why during midday, photosystem 2 activation outpaces photosystem 1. And that leads to an accumulation of plastoquinone. And that's bad because we want to keep this whole cycle going or else it'll... And so what I wanted to talk about is this like cascade compensatory mechanism that plants have and when the PQH2 accumulates. So we have here, PQH2 accumulates, it activates our protein kinases, and the protein kinases are going to phosphorylate our light harvesting complex, which you can think of these as our antenna molecules. Remember from this diagram, we have these antenna molecules that will transfer the electrons to the reaction center, our photosystem 2, photosystem 1. That's exactly how they act here. And we can phosphorylate their threonine residues. Threonine residues. That's very key. Remember the amino acid threonine has an OH group on the R. So we can phosphorylate that, and it'll basically spatially move it towards photosystem 1. So we see that the protein kinase will help move this light harvesting complex closer to photosystem 1, and that prompts its activation even more. And this occurs until the balance is restored. And once the balance is restored, we're going to start using protein phosphatases, which get rid of this phosphate group and move the light harvesting complex towards the oppressed areas. So just to give an overview of photophosphorylation, so 6 protons are transferred each time from a single O2 molecule. From one water molecule coming in. And I think the best way to actually interpret this is from a single O2 molecule, right? As you can see, this picture is just showing you half. But from a single O2 molecule, we're going to produce 3 ATPs because there's going to be 12 protons moved across the bilayer. So what does that tell you? Is that approximately 4 protons are needed for producing 1 ATP. Now, think about that and compare it to ETC. ETC, we know, requires 3 protons to form the 1 ATP. This one requires 4, so it's less efficient. And that's because there's a dissipation of charge in the membrane between the lumen and the thylakoid membrane and the stroma. And that means it's because electrogenic transport is not as effective. That's the whole reason why. And in terms of that single oxygen molecule, it's just double of the protons. So again, this is half, so we would have to double everything that's here, but essentially we would have the 2 coming in, 2 protons that are evolved from the oxygen evolving complex right here. We would also have 2 that come in through photosystem 2, because that's what reduces our PQH2. And then the PQH2, the cytochrome B6F, will basically pump 2 more. But these 2 can basically be formed together to form the 4, as seen over here. When I was mentioning earlier, 4 protons per pair of electrons. It's because 2 of them come from the Q cycle, and then the other 2 come from the water that's just not shown here. So don't get confused. Hopefully that makes sense. And yeah, that's just an overview. So something I want to just emphasize is that just remember this stuff in bold, especially when you're doing calculations, because this exam, they will test you on calculations. From a single O2 molecule, 3 ATPs are produced, and 12 protons move across the bilayer, right? So just remember those numbers, like write them down, memorize them, like burn them in here solidify them in here despite all the enzymes and everything you have to memorize. But that's something that you should definitely memorize. I don't have a helpful mnemonic. So it's something that keeps you grounded when you're doing the calculations. And it's best to obviously understand why these numbers come from and where they come from in the first place. So just keep that in mind. So moving on to the Calvin cycle, right? So we just to orientate ourselves, we produce ATP from the proton gradient. We produce NADPH from the non-cyclic reactions. What do we do with those? We use the Calvin cycle. That's exactly what ends up happening. So in this case, it occurs in the stroma. So remember, the ATPs that we produce, this is the handy thing. We don't have to transport them using the electrogenics. Remember that in ETC, we have to normally pump out our ATPs and bring in ADPs, like it was that like antiport or whatever. Well, we don't have that here because the ATP is already in the stroma. So we don't need that. That's why it's less or the electrogenic transport is not as effective, because we don't have that ATP-ADP switching. Sorry, I didn't mention that before. But essentially, the Calvin cycle is going to use the ATP and the NADPH to convert our carbon dioxide into a glyceraldehyde 3-phosphate, which is a sugar that we can use. But I will say, 1 carbon dioxide will not produce our 1 glyceraldehyde 3-phosphate. No, it requires 3, and we'll explain why. But I really, really, really want to emphasize this bottom part, which is for every 3 CO2 molecules that are fixed. That's why this is in parentheses. A lot of these are in parentheses because it's accounting for the 3. When there's 3 CO2s that are fixed, we form 9 ATP, 6 here and 3 here, and then we form 6 NADPHs. And we consume 6 NADPHs, and 1 G3P is produced as a result. So we'll explain this in detail. And one key intermediate I want to talk about is the ribulose 1,5-bisphosphate, which is regenerated using the ATP and the 5-glyceraldehyde 3-phosphates. And just to make this make sense, there are 5 carbons here, right? And we're multiplying by 3. That is 15. Over here, we have 3 carbons, and then we're multiplying by 5. Not 6, because remember, 1 leaves. But 3 times 5 is 15. So carbon to carbon, like numbers, they match. And so that's why we need to use 3 in order for this Calvin cycle to... So now moving into each stage, we're talking about stage 1, carbon fixation. We're converting the CO2 into 3-phosphoglycerate. So we're basically going to multiply everything by 3, but in this diagram, we're showing 1 at a time. So this is 1 ribulose 1,5-bisphosphate, and this is CO2. And this is Rubisco. It's an enzyme that is so slow, but and the only way to compensate for that is to just have a huge quantity of them. So even though it's slow, we just have a huge quantity, and we can actually make a sustainable amount of this, whatever molecule it's called. I don't even know. But Rubisco has very similar activity to biotin. It is not the same, though. Biotin only binds to CO2 to form carboxylation reactions. But Rubisco, you can think of it as carboxylation, but remember, carboxylations are typically found at the ends of the molecules, right? In this case, it's in the middle. So this is not a carboxylation per se, like in the strict sense. What this is it's just incorporating the CO2 into the middle of it. And one thing I will note between Rubisco and biotin, the difference is that Rubisco can combine either the CO2 or the O2. And O2 is photorespiration, which we'll talk about at the very end of this slide set. And the product at the end of carbon fixation is our 3-PGA, 3-phosphoglycerate. And again, there's a hydrolysis. So 5 plus 1 forms 6. We divide it by 2, and that forms our 2 3-carbon molecules because of the hydrolysis. That's our first step. And remember, this all occurs times 3. So 3 of these are going to combine with 3 of these, and then we'll form our 6 3-phosphoglycerates, because there's 2 here. 2 times 3 is 6. So then moving on to stage 2, we're going to convert our 3-phosphoglycerate. This diagram only shows 1 at a time. Remember that. So we're going to convert our 3-phosphoglycerate into glyceraldehyde 3-phosphate. And the way we do so is we phosphorylate it to form a 1,3-bisphosphoglycerate. And from this 1,3-bisphosphoglycerate, we're going to reduce it with NADPH, and that'll form our glyceraldehyde 3-phosphate. And something I want to emphasize is that this is happening 6 times. So that's where we get the 6 ATP and the 6 NADPH. And these 2 energy molecules are generated from the light reactions. Without the light reactions, we don't have this occurring. And only 1 G3P, because we're going to form 6, only 1 of them is allowed to leave the cycle. And the other 5 are used to build up the 1,5-bisphosphate, ribulose 1,5-bisphosphate, sorry. And yeah. So moving on to stage 3. This is the regeneration of ribulose 1,5-bisphosphate. Let's orientate to the diagram. I'm not even going to bother with this stuff. This will make sense in a little bit. So let's look at the red rectangles. Those are our substrates. Now I want you to note the number of red rectangles. Don't worry about these intermediates, these enzymes. No, you do not need to memorize it. This would be crazy if you had to know xylulose 5-phosphate, and then what catalyzes between steps 7 and 8. Like that, no. Don't worry about that. Just count the number of rectangles. There's 1, 2, 3, 4, and 5. 5 red rectangles, right? Well, that corresponds to the number that we need to regenerate the ribulose 1,5-bisphosphate. How many? Now, the purple rectangles, these magenta ones, are corresponding to the products. There's 1, 2. Well, you may say, Alex, there's 2. Where's the third one? Well, it's kind of hidden in this diagram. That's why I don't really like it, but this is the one that they use in the textbook or in your slides. But essentially, there's step 9 that occurs twice here, 1 and 2. And no, you don't have to memorize step 9 being special. Just count this rectangle as 3. Now, what I'm trying to get at is that we need these 5 to regenerate the 3. And so there's math to this. If we have 5 3-carbon molecules, right, and we multiply it 5 times 3, that's 15. Whereas our products are 3 5-carbon molecules. So 3 times 5 is 15. So they match. Now, in order to get from ribulose 5-phosphate, which a lot of these are ribulose 5-phosphate, we have to phosphorylate them. And we form 3 of these. So that's why we use the final 3 ATPs. And the net energetics of the Calvin cycle is that we use 9 ATPs and 6 NADPHs. So that is what I wanted to emphasize. And that is where we get to this beginning part where, again, we use 6 NADPHs and then 9 ATPs at the very end between stage 2 and stage 3. That's it. Now, this slide is talking about, so what if you make that 1,3-glyceraldehyde 3-phosphate, right? Right here. We have one, and it can be used for energy production. It could be used for sugar synthesis. It could be used for a variety of things. Well, remember that this is in the stroma, right? The Calvin cycle is occurring in the stroma. How does it get outside of the stroma, out of the chloroplast, into the cell, right? Cytosol in the stroma. That's what I want to emphasize with these red rectangles. So how does a glyceraldehyde 3-phosphate leave? That's what this transporter is telling you. And this is an antiport. So just to orientate yourselves, we see that this, what I'm circling right now, the 3-phosphoglycerate, the 1,3-bisphosphoglycerate, and the glyceraldehyde 3-phosphate occurring in the stroma is step 2 of the Calvin cycle. So this is step 2. And glyceraldehyde 3-phosphate will undergo an auxiliary reaction where it'll be converted into dihydroxyacetone phosphate through this triose phosphate isomerase. Again, same enzyme as glycolysis. And this enzyme is going to form DHAP. That's why I like to call this molecule. And DHAP will go from the stroma to the cytosol, and it'll be an antiporter to inorganic phosphate. Now, inorganic phosphate is going to go from the cytosol to the stroma, so they always go opposite to each other. And this is, and dihydroxyacetone phosphate will then be converted to glyceraldehyde 3-phosphate. And then now it can do whatever it wants. But sometimes, we need to bring in and increase our concentration of 3-phosphoglycerate or all this stuff, and we need to move stuff inside, right? Because we need to move inorganic phosphate out. I don't know. But the cycle can continue and move its way back in through 3-phosphoglycerate, in which we form NADH and ATP. And one thing I want to point out is that the stroma, again, this is stage 2 of the Calvin cycle, we use NADPH. Whereas on this side, we're using a different enzyme. We're using glyceraldehyde 3-phosphate dehydrogenase, which forms our NADH, right? And so you would do this reaction if you're doing glycolysis, right? This is like a going down glycolysis towards Krebs. Whereas this glyceraldehyde 3-phosphate could also just be used for gluconeogenesis, glycogenolysis, all that stuff. It could be used for the auxiliary reactions, the anabolic reactions. So there's this now diverge, and I recommend that you build this almost like into like a master document or like a master like pathway with like the organelles labeled where stuff is occurring inside, outside the mitochondria, cytosol, all that stuff. So one thing I want to iterate between this interplay between the light-dependent reactions and the light-independent reactions, so that photosynthesis and Calvin cycle, is that the Calvin cycle is indirectly activated by light. Why is this? Because the pH in the stroma increases from 7 to 8. In Rubisco, you remember like we talked about the enzyme optimal activity stuff? Well, Rubisco's optimal activity is at pH 8. So when light is going through, remember, when light happens, we have photosystem 2 being activated. It basically activates cytochrome B6F complex, and that will pump protons into the thylakoid lumen. And because that happens, the stroma is going to become more positively charged because there's less protons. It's going to be less positively charged because there's more protons, and therefore increase the pH from 7 to 8. And then the second thing, and this is memorization, I don't know why, but magnesium also increases the concentration from 1 millimolar to 6 millimolar. And that could just be because the protons are going inside the thylakoid lumen, and then there's a negative charge, so the magnesium is drawn towards it because of electrostatic forces. That's just me conjecturing. I don't actually know the answer. And then in light, the Calvin cycle activity increases more than 100-fold. And this makes sense, though, because you can't have the light reactions without light, and if you don't have the light reactions occurring, then you won't have sufficient NADPH and ATP. So it just wouldn't make sense to go through the Calvin cycle if you don't have the energy in the first place. And now moving on to photorespiration. Remember when I talked about Rubisco being different than biotin, and Rubisco can incorporate oxygen with CO2? That's exactly what this is. It's incorporating the O2 rather than the CO2. It does a misincorporation. And so this is in C3 plants, and we have photorespiration, where it's a wasteful side reaction. No, you don't need to know every little detail about this like pathway and salvage pathway. What you need to know is oxygen gets incorporated, and you form a 2-phosphoglycolate. Well, this is not the molecule that we use for stage 2 of the Calvin cycle. So what ends up happening is it goes through the salvage pathway through the peroxisome, mitochondrion, and you end up using an ATP. That's it, to form our 3-phosphoglycerate. So this is what we can use. So we went through this alternative pathway, and we form our 3-phosphoglycerate, but at the cost of 1 ATP. So this is very energetically costly, and that's all you need to know from this. You don't need to know glycolic acid oxidase. Like, see, I even had to like take a moment to see if I actually knew how to pronounce it. Like, you don't need to know this stuff. You just need to know that it forms a 2-phosphoglycolate, and that's gonna go through this long salvage pathway through different organelles, peroxisomes, mitochondria, because that's where these special enzymes are, and then eventually it'll become our 3-phosphoglycerate. That's literally it. So now moving on to alternatives to avoid photorespiration. So now we're talking about C4 and CAM plants. So C4, basically they have 2 different compartments, and they do this on purpose. So rubisco is going to be in the bundle sheath cell compartment, which is far away from the oxygen where it can come in. So the oxygen can go in the mesophyll cell, but it can't pass through into the bundle sheath cell, and only CO2 can. And it's through this like organic acid cycle, whatever. You don't need to know that. Just know that CO2 can enter a different compartment, O2 cannot. And this spatially separates CO2 and O2. Well, okay, no, I guess you do have to know CO2 is fixed into oxaloacetate. Oxaloacetate moves into bundle sheath. I even forgot, so it's not important for the MCAT at least. So then for the second part, right, we have CAM plants. And this is not spatial. This is temporal, meaning that this happens at night or at day. So what ends up happening is the stomata of the plants open at night. And this allows CO2 to come in. And it gets fixed into oxaloacetate. And it'll stay like that for the night. And during the day, then the oxaloacetate will basically separate the CO2, the stored CO2, and the CO2 will be used in rubisco to undergo the Calvin cycle. And we need light for photosynthesis to occur. So that's exactly why we store it in oxaloacetate first at night, and then we utilize it during the day, because we now have the light to power it through. And again, these plants are in hot, dry climates. Same thing with C4 plants. And then that's just the photosynthesis overview. I mean, hopefully you guys actually like feel like more comfortable with this material. I know it's a lot, an hour and a half, bro. I take 40 minutes to do this, but essentially, hopefully you guys feel comfortable about this material with the light dependent reactions and the light independent, and how they interplay with each other, and how they're connected in terms of energetically activation and inhibition, all that stuff. And so again, you're taking the electrons from water and light, because the light is what's powering it, and then you form your high energy molecules, and then that's what powers your reduction of carbons and carbon fixation. And you're going to take a fully oxidized carbon and reduce it. This is a fully oxidized, meaning that we know, just a little orgo interlude, just because I'm extra like that. We know that we're talking about a primary alcohol, right? Primary alcohol is just an OH attached to a methyl. And then we can oxidize that into an aldehyde. We can also oxidize it to carboxylic acid, but we can't go any further in terms of oxidation. That's the same principle here. So we have CO2 is fully oxidized. Then forming the CO is just your carbon, oxygen, carbonyl like double bond. You can think of it as like an aldehyde, formaldehyde. And then from there, you also form a COH, which is just your methanol, I think. Yeah. See, like I'm just trying to pull these off, but the point is that you're taking a fully oxidized carbon, like truly fully oxidized. You can't go anymore, and you're reducing it, going the opposite direction against the grain. So hopefully that made sense. If not, just ignore what I just said. Moving on to lecture 12. Let's power through this, bro. I got this. Okay, lecture 12. Gluconeogenesis and glycogen metabolism. So what is gluconeogenesis? It's in the name. Gluconeogenesis. Neogenesis. We're forming new glucose. That is it. From molecules at the bottom. So generating of glucose from non-carbohydrate carbon substrates. So it's an anabolic process. You have lactate, gluconeogenic amino acids, pyruvate, TCA intermediates that can serve as your starting substrate for gluconeogenesis. However, triacylglycerides tags cannot be used to generate glucose in vertebrates, and that's because we don't have the glyoxylate cycle. That's the only reason why. What I want to emphasize is that we cannot use acetyl-CoA for gluconeogenesis. It is not glucogenic. Why is that? Because I want to emphasize this again. I emphasized it in my lecture 9. Bro, a second fire truck? I want to emphasize this in lecture 9, from lecture 9 with the Krebs cycle. When acetyl-CoA goes in that cycle, it is exactly the same. Those are the carbons that are going to become our carbon dioxide that leave. In no instance did the acetyl-CoAs become our endoplerotic reactions. They don't get added to the intermediates, like in terms of replenishing. They will eventually come out as the carbon dioxides. So that's why I want to emphasize is that acetyl-CoA cannot be a part of gluconeogenesis. And gluconeogenesis occurs mainly in the liver, and this is because we want to prevent hypoglycemia in your body. So you don't want to run with low blood glucose levels, because we know glucose is our main energy molecule, right? And so we don't want that. And our brain primarily uses glucose as its primary metabolite, and then it moves on to ketone bodies, all that. We'll talk about that in the next lectures, in the next exam, so don't even worry. So gluconeogenesis is not just a reversal of glycolysis, meaning that glycolysis, we know that 7 out of the 10 steps are reversible, but 3 of them are not. And those 3 are going to be exactly what we need for extra bypass reactions in gluconeogenesis, meaning that right here we see that they're highlighted in red right here, and they're actually just given names. So here we have hexokinase. It's irreversible, so we have to use a different enzyme for gluconeogenesis. We have to use glucose-6 phosphatase. And then this one we have PFK1, where we use a fructose 1,6 bisphosphatase. And then down here we have pyruvate kinase. That's irreversible, so we have to do a 2-step bypass reaction here as well. So instead of doing like a 1 enzyme to here, we have to do a double bypass. So we'll go into that in detail. And due to these glycolytic reactions being highly exergonic, it's what causes them to be irreversible in the first place. And we have to use different enzymes, as I talked about. And there are a lot of points of regulation among these 3 irreversible steps. So there are plenty of options of choosing glycolysis or gluconeogenesis. And we'll talk about it, and I will try my best to break them down, because I know it is very confusing to interrelate all of them. So this is another example of the delta G's. No, you do not need to memorize negative delta G's. I know I was making a joke about that with a friend of mine, and I was like, yeah, you have to memorize all delta G's. And it's like, no, I'm just kidding. You don't. You just need to know which ones are irreversible and exergonic. That's pretty much it. So talking about bypass reaction 1. Remember that we're starting from the bottom, from the very start of gluconeogenesis, which is pyruvate. So we know pyruvate kinase is exergonic, and we need to bypass it using a 2-step enzyme. So that's what we're going to talk about. And there are 2 different conditions here. So that's what these 2 slides are. So there's pathway number 1, where tissues lacking lactate. I mean, in the liver specifically. So in the liver, there's a different pathway for this in other tissues. So pyruvate is transported into the mitochondria. What is it transported through? The sym port. Remember that pyruvate proton sym port? That's exactly what brings it in. So pyruvate is turned into oxaloacetate using pyruvate carboxylase. This is a new enzyme, not in the Krebs cycle, and it's called pyruvate carboxylase. And before I even jumped the gun, what is a carboxylate? What would be like a coenzyme that is used for this, right? And I'm trying to allude to think of Rubisco. Something I said that was similar to Rubisco, but it's not the same. It's biotin. And so biotin is going to be our coenzyme that's going to add the CO2 onto pyruvate to form our. So the reason why we do this pathway in the first place is because our pyruvate carboxylase is not found in the cytosol. So we have to go in the mitochondria to convert it into oxaloacetate, and then from there we can use oxaloacetate and convert it to PEP. If we were able to do pyruvate into oxaloacetate in the tissue. So we were able to take pyruvate and convert it to oxaloacetate in the cytosol, then we wouldn't need to do this whole transport thing with the malate dehydrogenase, all this stuff. We wouldn't. And this uses, again, the malate aspartate shuttle to move malate from in the mitochondria matrix to the cytosol. So again, there's a lot of interplay with the lot. And then eventually oxaloacetate is going to be converted to PEP through this PEP carboxykinase. And carboxykinase is different than a carboxylase. Carboxykinase will use a GTP and essentially get rid of the CO2 to form our next 3 carbon molecule. Sorry. Something to emphasize is that this is using the oxaloacetate to PEP is using cytosolic PEP carboxykinase. And cytosolic NADH is needed for gluconeogenesis for this. So that's in the liver. Now let's say pathway 2, where it's lactate generating tissue, like in the skeletal muscle, right? Skeletal muscle can also do this, but they also can generate lactate as well. So with this specifically, we're talking about instances where we have low blood glucose levels in the surrounding skeletal tissue area, and we generate a lot of lactic acid, right? This is how we break down lactic acid. So lactate will essentially undergo this reaction with lactate dehydrogenase, and it'll turn into pyruvate. Pyruvate goes through its symporter with the proton and go into the mitochondria and react with pyruvate carboxylase, which is only found in the mitochondria, and it'll turn into oxaloacetate. Now, oxaloacetate, instead of turning into malate and then being exported out, we have mitochondrial PEP carboxykinase for these types of tissues. So that you have to know that distinction between your liver, where the PEP carboxykinase is in the cytosol, and then in your skeletal muscle, where the PEP carboxykinase is in the mitochondria. So you do have to know that distinction. And so, of course, they use the same like substrates, they use the same energy molecules, all that stuff. They just occur in different locations. There's this spatial awareness that you should have in where they're found. And so once it's converted to PEP, PEP can be transported out through a transporter. Do I know? I don't know off the top of my head, but there is a transporter that moves PEP out. And then again, it uses GTP, ATP for these two enzymes. So talking about the pyruvate carboxylase mechanism, it's all here written in the notes, but the ATP that we use is going to, because it's not just carbon dioxide, it's our bicarbonate here, and it's going to get fixated into or converted into carbon dioxide using ATP. And that's what ends up happening here, and it binds onto biotin. Now, biotin will rotate itself and bring carbon dioxide closer to pyruvate, and pyruvate does its thing where the oxyanion kicks back in and then essentially grabs onto the carbon and carboxylates it, does all that. And essentially, what ends up happening is we form oxaloacetate, and that's what this is, essentially, this mechanism. Now, moving on to PEP carboxykinase, the important steps to know are arrow 1, arrow 2, 3, and 4, and so we'll go through this shortly. Arrow 1 is where we have the carbon dioxide, that negative charge, kick back, and it's going to break this bond, and what ends up happening is we're going to use another, like, ATP equivalent. Instead, we're using GTP, and it's going to break this bond, and our negative charge on, or our carbonyl group is going to grab onto one of the phosphates. And now we end up forming phosphoenolpyruvate. And that's basically the mechanism. It's like a 4-step mechanism, so definitely be familiar with this as well. So regulation of pyruvate carboxylase. So this is the bypass reaction number 1, essentially. So pyruvate carboxylase is actually regulated by acetyl-CoA. So acetyl-CoA does play a role in gluconeogenesis. It doesn't form the glucose, but it does regulate it. And I'll explain why. So when there's high acetyl-CoA, that means that the citric acid cycle is slowing down. And that's because we treat acetyl-CoA as a high energy molecule. So we don't need glycolysis, TCA, ETC to continue if we have a high concentration of this, right? It's being backed up because this citric acid cycle is being inhibited by, high energy molecules and like ATP, NADH, because we have a high surplus of them. And because that's being inhibited, this backs up, right? And the acetyl-CoA is then saying, okay, we're backed up right now. That's because the citric acid cycle is not going. So we're going to stop glycolysis or stop the pyruvate dehydrogenase complex from working, and we're going to stimulate pyruvate carboxylase to convert the pyruvate into oxaloacetate. And so that's why pyruvate dehydrogenase complex is inhibited in this, and pyruvate carboxylase is activated. And then moving on to bypass reaction number 2. So now we're moving up to, if I were to go back to the slide, we're moving up all the way to basically the top of glycolysis again. So now we're talking about this 3rd step in glycolysis, where we convert fructose 6-phosphate to fructose 1,6-bisphosphate. Now we're doing the reverse, but with a different enzyme. So we have fructose 1,6-bisphosphate converting into fructose 6-phosphate. So what ends up happening is we need to use water to hydrolyze one of these phosphate molecules, and it's going to be ding, 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 number 1 that we're going to hydrolyze, and we're going to form this inorganic phosphate, and it does not require nor generate ATP. So you may say why doesn't it go in the reverse direction? I don't know. And the reaction is not exergonic enough to add a phosphate group to ADP. I think that's why it doesn't just go in the reverse direction. So that's why we need this different enzyme. Oh yeah, and also the delta G's are listed here, so you can convince yourself of why it's not able to. So moving on to bypass reaction 3, glucose 6-phosphate to glucose. So this is the last step of glycolysis, the first step of glycolysis, last step of gluconeogenesis, where we're going to convert a glucose 6-phosphate into glucose. So we, again, hydrolyze it, and we need another another enzyme, and it's called glucose 6-phosphatase, where we bring in a water, we hydrolyze it. Again, it doesn't use or generate ATP, and that's because the reaction isn't endergonic enough to phosphorylate ADP. So we're moving all the way back up to the top, and I want to clarify this point of reaction is not exergonic enough to add phosphate group to ADP. So what I wanted to emphasize is that you may say, well, Alex, I'm looking at the delta G's here, and I see that they are negative, right? And actually that this one is much more negative, meaning that it should be. Well, this is standard conditions, and we know that in the cell it is not standard conditions. So I know it's not demonstrated here, and I wish I diagram had the actual values rather than not the standard values. But just know that this is not exergonic enough to basically go in the reverse direction. There's not enough energy. So that's why we don't use or generate ATP. And then moving on to this one as well, we see that in bypass reaction 3, this one, the standard values are more positives than the glycolysis direction. So it's not powerful enough to just go and add the inorganic phosphate to the ADP to form ATP. So something to keep in mind. And gluconeogenesis overall is very, very expensive. So essentially right here, for each pyruvate, or for the 2 pyruvates that we have to form our 1 glucose, we see that we have to take 2 ATPs here, and we have to take 2 ATPs here. So that's the 4, and then our 2 GTPs are right here. And then we also have to use 2 NADHs for the glycerol 3-phosphate dehydrogenase. And then from there, we end up forming a glucose. And it's highly regulated to prevent a futile cycle. Again, it's very expensive, so we need to have this extremely regulated. ATP and AMP are key regulators of this. ATP is high energy, meaning that let's do it. We can do this. We can expend energy. ADP and AMP favor catabolic. It's just saying, hey, it's saying, hey, we need energy. Like, we cannot do gluconeogenesis right now. We can't supply enough power to it. And then the glucose sensing F2BP, which we'll talk about in a little bit, will also be used to regulate these processes. And the rationale, and this is distinct from high energy. This is different. So F2BP, do not equivalate it with ATP or ADP or AMP. Do not combine them, because that's going to get messy. F2BP is just a glucose sensor, meaning that if there's high glucose, there's high F2BP. And you won't want to make more glucose even if you have the energy. So like, if this is already high concentration, it doesn't make sense to make more, because why? Instead, what will end up happening with F2BP is it's going to be a key regulator to say, hey, gluconeogenesis, we can't do this because we want to get rid of the high glucose concentration. We're going to stimulate glycogen synthesis. That's what it's used for. So keep them separate, and we'll talk about this more in depth. So talking about F2,6BP, or fructose 2,6-bisphosphate, we're going to use different isoforms of PFK and FBPase. So at PFK, we know PFK1, and this is just an isoform of it. And the reactant is fructose 6-phosphate, the same as PFK1, but it's going to form fructose 2,6-bisphosphate. And then for the breakdown of fructose 2,6-bisphosphate into fructose 6-phosphate, we're going to use fructose 2,6-bisphosphatase, which is essentially just an isoform of this one, fructose 1,6-bisphosphatase. And we are basically taking away the phosphate group from number 2 to form our fructose 6-phosphate. And from there, we can either go through glycolysis or go through gluconeogenesis, one of the two. And again, our F2,6BP acts as a glucose sensor. And this is going to this will form if we have high levels of glucose. It has nothing to do with energy. It just has everything to do with glucose. And so this is what I want to talk about in terms of regulation of PFK1 and FBPase -1. So this is what I want you to orientate yourself, because I know this is extremely confusing. Let's talk about the energy. We will not talk about the F2BP yet. So with PFK1 and FBPase1, for glycolysis and gluconeogenesis, we're talking about energetics. We are not talking about glucose. We're talking about energetics. So PFK is glycolysis. The only time we need glycolysis to occur energetically, not glucose concentrations, energetically, is when we're in low energy states. We need to develop energy. We need glycolysis to happen. So what's going to stimulate it? ADP, AMP. Those are low energy molecules. It's telling the cell we need glycolysis to occur. We need energy. We need to harness it from the glucose. So that's what it stimulates it. And it's in PFK, glycolysis is inhibited by ATP and citrate. ATP is a high energy molecule. If we have a lot of high ATP, we don't need to create more of it. And same thing with citrate. Citrate gets backed up when Krebs cycle stops going because of NADH and inhibition there, and then it basically backs it up and clogs it. And the citrate will then say, hey, we have high energy, we're clogged, we don't need more. This is energetically. This has nothing to do with glucose concentrations. It has nothing to do with that. This is all energetics. And then we'll skip the F2,6BP being stimulated, and we'll talk about other things. So with fructose 1,6-bisphosphatase, now we're talking about gluconeogenesis. So gluconeogenesis will essentially be stimulated when we have high energy states, right? So if we have ATP, it's going to stimulate it. If we have low energy molecules, it's going to say, hey, we don't have the energy to do it. We're not going to be able to do gluconeogenesis. We don't have the power to do so. So that's what AMP serves as an inhibitor of this gluconeogenesis pathway. Now, I want to talk about F26BP. This is different. This has nothing to do with energetics per se. It is not necessarily solely energetics. So separate the ideas and this will become crystal clear. F2BP is a glucose sensor. When there's high glucose, this starts forming. When this forms, then we can stimulate glycolysis. This has nothing to do with energy, again, because if you think about energy, then you'll think, oh, well, if we have high energy, why would it stimulate glycolysis? Separate them. F26BP is telling the cell, we have high glucose, we need to get rid of it. And one of the ways is glycolysis. Another way is glycogen. And we'll talk about which way is preferred. That's where we have the interplay of energetics and this F26BP. We'll talk about that. So F26BP will stimulate glycolysis when we have high glucose. It will inhibit gluconeogenesis because we don't need more glucose. We already have a high concentration of it. So it just doesn't make sense. Even if we have the ATP for it, it's going to inhibit it because we don't need it. And what will end up happening? Let's talk about these alternative scenarios. Let's do this interplay now. Now that we have separated the ideas, let's talk about it in relation to each other. So let's say we have high energy, but we have high glucose, right? We know ATP inhibits PFK1, right? But we also know high glucose will stimulate it. So you may say, well, Alex, what's happening here? Well, what's happening is we have high glucose and we need to get rid of it, but we have high ATP, so we can't do it through glycolysis. Glycolysis says, we're done, we're not going. So what do we do with the glucose then? If we have a high glucose concentration, we're not gonna go through glycolysis, we're gonna go through glycogen synthesis. That is your answer. That is exactly what's gonna end up happening. And so what we don't see here is that it's stimulating glycogen synthesis, F26BP. So that's the first scenario. So if you have low glucose, obviously you want to form gluconeogenesis, right? If you have low glucose, you're going to want to form more. So gluconeogenesis is the pathway that we're going to want to stimulate, right? That's why we don't see this inhibition if we have low glucose. This is not going to be there, so therefore you're not inhibiting this pathway. Now, when we have, again, the high glucose, high energy, we're not going to get rid of this glucose concentration here. It's high concentration. We're not going to get rid of it through glycolysis because it's inhibited by ATP and citrate. It's going to go through the glycogen synthase pathway. And that's what's not shown here. But that's what I want to get across is that keep these ideas separate, these regulators, because F26BP has not necessarily anything to do with energy. It just is about glucose itself. Whereas ATP, ADP, AMP, citrate, those are all metabolites of glycolysis, Krebs cycle, etc., cellular respiration. And yeah, this is just exactly what I was just telling you is fructose 1,6-bisphosphatase is also stimulated by glucagon. What is glucagon? Glucagon is a hormone that is secreted that tells you, hey, we have low glucose levels and we need to generate more. And so gluconeogenesis is going to be stimulated from all the cells so we can generate glucose and to transport it into the blood. So we maintain a stable blood glucose levels. And again, it's an allosteric regulator, it's not intermediate, and it acts as a glucose sensing molecule as well, along with fructose 2,6-bisphosphate. So now I want to talk about the regulation of PFK2 and FPPase2. And just to orientate ourselves, PFK2 is what forms our fructose 2,6-bisphosphate, and FPPase is the one that takes this away and forms fructose 6-phosphate. So that's what I wanted to say, and that this is a dual functioning enzyme, meaning that there are two domains on the same peptide, and only one is active at a time because they're physically linked. And so what this means is that PFK2 is inhibited by glucagon and CAMP. Now, why is that? So we see PFK2 is going to essentially, and so essentially PFK2 is going to form our fructose 2,6-bisphosphate, which is our glucose sensor. This only forms when we have high glucose levels. So if we have glucagon present, then we're saying that, hey, cell, we have low glucose levels, we have low CAMP, and CAMP is also saying we have low glucose levels. So stop making F26BP because you're tricking yourself that you have a lot, but you don't. And so what ends up happening is glucagon and CAMP, by mechanism, they will basically stimulate CAMP dependent protein kinase. This is all details, but all I'm trying to say is that the PFK2, when it's active, it's just a hydroxyl group. When PFK2 becomes inactive and the other part of it, FPPase2, becomes active down here, it's phosphorylated. And glucagon and CAMP will activate this kinase right here, and it'll phosphorylate our dual enzyme here. And that will stimulate the FPPase2. And because it's stimulating FBPase2, that's technically inhibiting PFK2. And so it's like a reverse, essentially. I don't want to confuse you. But one thing that does make sense is PFK2 is stimulated by insulin. When is insulin secreted? Insulin is secreted when we have high blood glucose levels by the pancreas, the beta cell specifically. And so when the insulin is secreted, it's going to stimulate our phosphoprotein phosphatase, which all it is we're taking our phosphorylated dual enzyme and we're getting rid of it. We're dephosphorylating it and forming our hydroxyl group, which then stimulates the production of F26BP. And that has its corresponding effects on gluconeogenesis and glycolysis. Insulin is saying, hey, we have high glucose levels. It's a glucose sensor, and we need to tell everyone that, hey, we have high blood glucose levels. We need to sequester the glucose from the blood and convert it either into glycogen or glycolysis, one of the two. And again, whether or not it goes down glycolysis is dependent if it's high, if the cell has high energy or if the cell has low energy, right? So something to keep in mind. And then fructose 2,6-bisphosphatase, FBPase2, is and it's the reverse now. It's inhibited by insulin because insulin stimulates the phosphatase and it prefers this PFK2 activation and FBPase2 inactivation. And so we'll dephosphorylate, and then it's stimulated by glucagon. The FBPase2 is stimulated by glucagon and CAMP because the kinase is stimulated, and that forms the phosphorylated version of this dual enzyme, and that will basically get rid of our F26BP, and that'll have its corresponding effects on gluconeogenesis and glycolysis. And now shifting to glycogenesis. So glycogenesis is generating sugar nucleotides, and we're not just going to add glucose together. That's not how it works. We need to form our UDP glucose for glycogen itself. That's the substrate for attaching them as like building blocks. We need to form this UDP glucose. And so there's a 3-step process to form UDP glucose. The first is we add glucose and ATP to form glucose 6-phosphate and ADP. The enzyme is hexokinase, or also known as glucokinase, depending on which part of the body you're in. That doesn't really matter. But then the glucose 6-phosphate, and again, this is also glycolysis intermediate as well. And also the pentose phosphate pathway intermediate as well. Just emphasizing that there's a divergence. And so it gets basically mutase, which just means that you're transferring the functional group from one place to another. And it's a phosphoglucomutase, meaning that it's a phosphorylated glucose, and that phosphate group gets mutated from carbon number 6 on glucose to carbon number 1. And then with this glucose 1-phosphate, it's going to combine with a UTP, which is a uridine triphosphate. It's a base that's used for RNA synthesis. And then it's going to combine with 2 inorganic phosphates to form a pyrophosphate and our UDP. And then it's going to combine to form our pyrophosphate and our UDP glucose, which can now be used for glycolysis. And the enzyme that catalyzes this is called UDP glucose pyrophosphorylase. And I put these in bold because you should know all the enzymes and how they work, and these are just pictures of them. And essentially why we use tag sugar nucleotides for biosynthesis. The reason why is because it's irreversible because of the pyrophosphate. It immediately gets hydrolyzed, so we're taking away the products. Again, very similar to some of the Krebs cycle steps, like going from oxaloacetate to malate to oxaloacetate. And then secondly is there's binding energy, which is favorable enzymatic interactions. There's reactivity with good leaving groups, bad leaving groups, and then separate pools because of, we're going through these 3 different steps. And this is just a picture of UDP glucose. So something I want to emphasize is the phosphoglucomutase mechanism. And I want to say at first, this is showing the reverse direction where I know here we're saying that it goes from glucose 6-phosphate to glucose 1. This is reversible, so it can go backwards as well. That's what this mechanism is showing. So I don't know why this is how your textbook presented it. It is what it is. So we have our phosphorylated serine residue, and this is going to donate our phosphate to glucose 1-phosphate, forming glucose 1,6-phosphate. So as you can see here, here's our phosphate. It's going to attach to carbon number 6 of our glucose 1-phosphate and form this bisphosphate glucose. And then from there, one of them is going to be donated onto that serine, and now we just did the full mutation. That's literally it. We add it to form this like bisphosphate, and then we take the other one away. And now glycogen synthesis, we have to prime it, right? So glycogenin is like essentially like our starting block to prime and generate new glycogen molecules to form those branches, those chains. And so it's a protein, right? And the process is the glucose residue from UDP glucose binds to a tyrosine residue on glycogenin, this protein, and then it adds a sequential addition of 7 more glucose residues, and its extension then is by glycogen synthase. So glycogen synthase does not do this process initially, it does the subsequent by adding it like Lego bricks. Glycogenin remains bound to the reducing end of the glycogen molecule. So the reducing end is over here, right here, and the non-reducing is on the other side. If I said that correctly, if that makes sense. And then, yeah, glucosyltransferase activity is the enzyme, and you should know this is the enzyme that adds the glycogen to the UDP glucose. And then from there, we have chain extending activity for the next 7 molecules. And then from there, we repeat for 6 times or whatever, and then we use glycogen synthase afterwards. So glycogen synthase and branch synthase, these are the 2 enzymes that allow us to form our glycogen polymers. So glycogen synthase needs primers. And so this is the reason why glycogen synthase doesn't react right away. It needs a primer of 8 residues before it can actually work. So that's why we use glycogen to prime and use the chain extending activity. So it transfers UDP glucose to non-reducing ends of glycogen branches. So here, as you can see, this is a new non-reducing end. Remember what reducing and a non-reducing sugar is. We're looking at the anomeric carbon. The anomeric carbon is essentially the carbon that's connected to 2 oxygens. And so here's carbon 1 that's connected to 2. It's already participating in a glycosidic bond, so it's not a free reducing end. And remember, we added all of this to the glycogen core, the glycogenin. So the reducing end is always going to be participating in glycosidic bond. And so there's never going to be a free reducing end at the other side of it. And so that's why we need glycogen synthase. That's why it doesn't just form spontaneously. We need this enzyme to help us in the first place. And glycogen synthase forms the alpha-1,4 linkages. And the glycogen branching enzyme, now talking about this other one, is where it transfers the terminal end of 6 or 7 residues from a non-reducing end and basically puts them on the same or different chain. So as you can see here, we have 1, 2, 3, 4, 5, 6, 7. We're going to cleave this bond. We're going to move it to our carbon number 6 of our wherever in this chain. It doesn't have to be the first one. It could just be later on. And as you can see here, it forms an alpha-1,6 branch. And again, the alpha is very important for this as well to memorize. So now shifting to glycogenolysis. So I broke down the word glycogen, lysis, so breakdown of glycogen. So glycogen breakdown. So we have glycogen phosphorylase that does this. What it does is it basically breaks off from the non-reducing ends and breaks them off like Lego bricks, one by one by one by one, and forms glucose 1-phosphate. And then all it needs to do is just convert to glucose 6-phosphate via the mutase, the phosphoglucomutase, and that's it. You're chilling. And one caveat about glycogen phosphorylase is it stops 4 residues before the branch point. So you count 1, 2, 3, 4, and it stops. Glycogen phosphorylase will not break any of these bonds. What ends up happening is we have a debranching enzyme, and there's 2 types of activities within this enzyme. There's a transferase and a glucosidase activity. So transferase basically will take the three glucose monomers from the non-reducing end and transfer them over to the ends of the chain, right? And what will end up happening is it removes the it'll shift those 3 to non-reducing, and we're left with our last one. And it'll resolve the alpha-1,6 linkage by this glucosidase activity, and it releases it as glucose, regular glucose, not glucose 1-phosphate. So that's a caveat that you have to know. And then from there, we just got rid of the branches, and then we can start breaking it down. And then this is the last slide, I'm pretty sure. Yeah. So regulation of glycogen synthesis and breakdown. So glycogen phosphorylase and glycogen synthase can be regulated. And so I will orientate you to these 2 diagrams. And one thing to note is that look at their OH residues on these enzymes and look at whether or not they're active when they're inactive or active when they're phosphorylated or hydroxylated. And that'll give you a big clue because phosphorylase is the opposite. It'll be active when it's phosphorylated. Glycogen synthase will be active when it's hydroxylated. So glycogen phosphorylase is active when phosphorylated, inactive when dephosphorylated, and it's activated by glucagon, epinephrine, increase in calcium, increase in AMP in muscle. So we'll stick with this right diagram, phosphorylase. So it's activated when phosphorylated, and so as you can see, we need to phosphorylate it using this phosphorylase B kinase. And what ends up happening is glucagon stimulates it, and epinephrine, calcium, and AMP and muscle can also stimulate it as well. Okay. So the best way to think about the regulation of glycogen synthesis and breakdown is what are they used for? Glycogen synthase, is used to build up glycogen, right? And phosphorylase is used to break down glycogen. So keep that in mind as we go through this. And also note that glycogen synthase is activated when it's hydroxylated. Phosphorylase is active when it's phosphorylated. So it'll favor one of the things. If we have overall gross kinase activity, it's going to phosphorylate our phosphorylase and phosphorylate our glycogen synthase. So we're going to favor this activity, the breakdown of glycogen. Whereas if we have our phosphatase activity being activated here, then we're going to favor the hydroxylated versions of these enzymes. And one of them is active, the other is not. So something to keep in mind. The glycogen phosphorylase is activated by glucagon, epinephrine, calcium, AMP, and all that stuff. Now, why is that? So glucagon, what does it mean? Hey, cell, we have low glucose levels. We need to get glucose. So, of course, glycogen phosphorylase, if it breaks down glycogen, it's going to be stimulated by glucagon. Because phosphorylase will simply say, hey, I'm going to work overtime and I'm going to break down glycogen so you can have free glucose molecules. That's what it's essentially saying. And so that's why it's activated. And then epinephrine will also activate it because epinephrine is like a fight or flight response. And one of the key features of fight or flight and adrenaline responses is that we get a surge of glucose levels in our bodies. That's because we need to pump the heart and all that stuff. And then increasing calcium, well, that is because when we increase calcium, that's usually when the muscles are working. And when the muscles are working, we need ATP. We need to generate that glucose right away. And we need to use that glucose for ATP synthesis. So glycogen is going to be broken down because that's a readily easy source to use. And then it's also going to increase in AMP because if we have a lot of glycogen and we have low energy, well, we would like to break some of the glucose from the glycogen and use it for energy to form energy. Now, glycogen synthase, it's going to be the opposite, right? And so this is a little bit more of a complex reaction. So it's activated by insulin, which makes sense. If we have insulin, we're telling the cell, hey, we have a lot of glucose in the blood. We need to get rid of it. And one of the ways that you can do it is through glycogen synthesis, glycogen synthase. So that's why it stimulates it. And then glucose 6-phosphate and glucose are also going to be activators of this because if we have high concentrations of these, then we either we'll go through glycolysis, which it already will, or sometimes we can just form glycogen just because of the higher sheer number concentration without looking at the energetics, right? And so one thing I want to emphasize is that none of these are really looking at energetics. It's looking more so at glucose levels. And that's really like how you should think of this instead of trying to interrelate energies with it. You can, but again, it goes back to the F2,6BP and the ATP and inhibiting glycolysis through the high energy, but also because you have high glucose, you need to stimulate glycogen synthesis. So that's like the one way to think about it, but I don't want to confuse you guys. And then the reason why insulin, in terms of mechanism of action, why it activates our glycogen synthase is because insulin inhibits GSK3. GSK3 is just a kinase. And remember, kinases phosphorylate things. So our glycogen synthase, when phosphorylated, is inactive. So if we stop this phosphorylation from happening, we're going to keep this prolonged activation. That's why it stimulates it. Insulin is inhibits an inhibitor. So GSK3 is an inhibitor of this glycogen synthase, but we're getting rid of this action. So it no longer can go this direction. And then also I want to say that glycogen synthase is inactivated by glucagon. And that makes sense. If we have low blood glucose levels, we don't want to form glycogen because that's just taking more away that could be given to the blood. And then same thing with epinephrine. It's a fight or flight response. We don't want to store stuff. When we're in fight or flight, we're breaking stuff down. We're not resting and digesting. I don't know. And then I have a bunch of practice questions here. And honestly, you can go. And so now, these are high yield topics that I think you guys should know. I'm not going to go through them because it's already been a 2-hour video so far of me recording. So definitely something for you guys to look at. And then also there's a lot of practice problems I put in here, like a lot. I mean, like a lot. So go through these through a slideshow. I'm hoping that some of these can help you guys with understanding and getting more practice questions. But yeah, if you guys have any questions, please use AdStem, email, all that kind of stuff. Come to my office hours review sessions, all that stuff. I'm going to be hosting it. And yeah, I'm really wishing you guys luck on this exam. Hopefully this video cleared up a lot of things. Obviously rewatch it, do all the good things. Do this. I literally walked in this room and this whiteboard was already full, but literally write stuff out from scratch, like glycolysis, okay. And then start writing your Krebs cycle too. And then write them in different compartments as well. What's a really neat trick, and I recommend you guys, if you really want to be solid, is to if you're using GoodNotes or Notability, I don't know if Notability has this, but on GoodNotes, use one of the expansive pages that is unlimited scrolling. You can go in any direction and just literally write all the pathways possible, everything, in all the transporters, everything. It's going to look convoluted, but the point is that you remember everything and you see visually in front of you how they interrelate, and then just label them different colors. Glycolysis can be red, Krebs is blue, TC is green, right? And then all your shuttles, you can put all your shuttles as purple, right? And they serve as shuttles. Then you can put, for example, gluconeogenesis as purple. Change the colors so that way you see how, these two paths interrelate here. And you're going to discover, a lot of these cofactors that can interrelate and whatnot, the reversible reactions. That's the best way to study for this type of exam if you're trying to consolidate everything. And truly, I do mean it. And if you have to look stuff up because you just don't know it first, because you're maybe making your first pass, that is okay. But honestly, you're going to get more out of that than just trying to go through rote memorization, Anki cards, and all that crap. So that's just my best advice I can give. And hopefully, yeah, I wish you guys luck on the exam. But sorry I got this out really late, but wishing you guys the best.